Hi biologists, welcome back. Today we're going to talk about cellular respiration, an oxidative, exergonic, and super cool process. So let's get started. Organisms, we know, have to capture and store free energy for use in metabolism. It's one of the big ideas for the AP curriculum. It's big idea number 2. Biological systems utilize free energy and molecular building blocks to grow and reproduce and maintain dynamic homeostasis. It's posted right in my room. So it's a big deal. A lot of the AP curriculum will come back to the same idea. So how do we do it? Well, we just looked at photosynthesis in class, and we know that autotrophs, things like plants, can capture free energy from their environment. Plants utilize sunlight. There are also chemosynthetic organisms, largely bacteria. So think about before plants came on the scene, life was established on the planet long before plants came, so in the forms of bacteria. So bacteria had to have a way in order to capture free energy from their environment in order for them to build, metabolize, and grow. And the way they did it was from using inorganic compounds in their environment, largely aquatic environments early on. So, and then once oxygen started to build in the atmosphere, we see the evolution of eukaryotes and plants, things that ultimately will utilize oxygen for cellular respiration. So just be aware that autotrophs are more than just plants. They're chemosynthetic organisms out there that live in the deep thermal vents, think where there's no sunlight and there's extreme temperature. But these organisms are still able to utilize their surroundings in order to run through their metabolisms, and it's done through taking in inorganic compounds. All right, heterotrophs. We are heterotrophs, and we need to capture free energy by consuming things that have eaten other things. So, well, we consume plants, that's a huge part of it, but and other organisms that have relied on plants as well, other heterotrophs. If you have a nice steak dinner, that cow relied on plants. So everything really goes back to the autotrophs. We need those plants in order to capture that sunlight and turn it into sugar. And ultimately that allows life to exist on this planet beyond plants. So heterotrophs, we need to take in those carbon-based compounds. Why carbon? Remember, carbon is the organic molecule. So all of our compounds, when we looked at our biomolecules, what did every single one of them have in common? Every single one of them is based off of carbon. When we looked at fats and carbohydrates, obviously, proteins and nucleic acids, all carbon-based molecules. So all of the molecules essential for life are based on carbon. Therefore, we need to consume carbon compounds. And we break them down through hydrolysis, and then we're able to utilize the energy that's stored in those bonds in order for us to grow, divide, send impulses, everything we need to do. So the energy stored in those carbon-based compounds allows us to grow and to live. The energy is stored as ATP. So, if we remember ATP, remember the analogy of the third kid on the bus seat. So you have this molecule. It has adenine, the nitrogenous base, ribose is our sugar, and the phosphate group. And by the time you add that third phosphate, it's a very unstable molecule, and there's lots of energy stored in those bonds, especially in that second to third bond. So breaking off that third bond releases that energy, and we are able to utilize it. It's nice because it's small amounts of energy. Why would we not want a compound that could hold a ton of energy? Well, if you had so much energy stored in that little bond and then it breaks, we could have things like spontaneous combustion happening. There's heat released typically, and thankfully for us, because we're endotherms, we need to generate our own heat. So we need this process in order to maintain our homeostasis with our own internal temperatures. Ectotherms rely on their environment. We are endotherms, endo-in, so we rely on internal temperatures to be set for us, and that takes a lot of energy. Thankfully, we don't have so much energy stored in a molecule where we spontaneously combust. It'd be bad. We're going to look at 2 different ways we can make ATP. Substrate level phosphorylation makes tiny amounts. Oxidative phosphorylation makes lots, and so we'll look at the 2. So the basics. Remember the good old, this hopefully goes back to freshman biology. We have glucose plus oxygen. We're going to use this oxygen to help us break down this glucose. And I'll show you how. This oxygen, what do oxygens love? They love electrons, and that's going to help us as we move through an electron transport chain. So we need that oxygen. It's going to help pull all of that energy forward so that we can create ATP. And we're going to oxidize that glucose into 6 different CO2 molecules. We'll let those go. We breathe those out. We also breathe out water vapor, and we thankfully make lots of ATP. Where does this happen? Mitochondria. Who does it? Everybody, all organisms, yes, even plants. Plants make sugar, then they have to use cellular respiration to break it down. So don't forget that little fact. Do prokaryotes? Prokaryotes don't even have mitochondria. How do they go through cell respiration? Well, they utilize their plasma membrane, and I'll show you how that's done. So, but even prokaryotes have to go through cellular respiration. The purpose, we need to make that ATP. It is our energy currency. It carries those small amounts of energy. So everything in our body, every time you move, every time a neuron fires, every time your heart beats, just to maintain a normal temperature and not, it's like 60 degrees in my room now. It's freezing. So, but thankfully, I am burning fuel and creating an internal body temperature. Here's the overview. We're going to look at glycolysis, which is our first step where we break down that glucose molecule initially. We break it down so it's small enough to enter the Krebs cycle, which happens in the mitochondria. In the Krebs cycle, we're going to release a lot of CO2, so we're getting rid of those carbons, we're oxidizing those and stealing those electrons away. The electrons are going to enter an electron transport chain and help power a proton pump, which will then make lots and lots of ATP through ATP synthase. Sound familiar? Hope so. Okay, so we're going to look at a couple ways that we can do it. We can do it aerobically. Aerobic oxygen, if you're doing some sort of aerobic exercise, you're typically breathing quite hard, and so that's the oxygen component there. And we produce more ATP. That's the advantage by using oxygen. We can produce a heck a lot of more ATP, and I'll show you the difference between the two. This happens in the mitochondria, so it has to. That's where the electron transport chain is. That's where we're making all the ATP, and that's where that oxygen is going to sit and pull all those electrons towards it. And the anaerobic doesn't utilize oxygen. So ana is no oxygen being used, and only a little ATP can be made. It's important to be able to have this function when you're in a fight or flight response and you're sprinting, or if you're just working out and you're sprinting, you can move into an anaerobic situation where you're burning more fuel than you have oxygen ready to utilize, and you build up lactic acid. But it is allowing you to get a little boost of ATP over and above what you would do when you combine it with the aerobic version of cellular respiration. Okay, so the 3 steps: glycolysis, Krebs cycle, and electron transport chain. I don't want to spend much time on this because we're going to go through all 3 points. But starts with the glycolysis, moves to Krebs, and then ends with the electron transport chain, which also is involved with oxidative phosphorylation. For your purposes, if you just put ETC, people will know what you're talking about, but understand that that's going to drive oxidative phosphorylation, which we'll talk about in just a minute. But let's start with glycolysis. So, in glycolysis, glyco means sugar. Remember our glycoproteins on the cell surface? They were important in cell-to-cell recognition and cell identification. Glyco is a sugar. So glycolysis is breaking of sugar, and that's where we start. So glucose comes in. It's a fairly big molecule at 6 carbons. We need to break it down initially. Glycolysis happens out in the cytoplasm. So because it's too big to get into the mitochondria, we break it down in the cytoplasm. And also this process can be both aerobic and anaerobic. So if you're going through anaerobic respiration, you never even make it into the mitochondria. This is running in the cytoplasm. This happens during glycolysis. Once it moves to Krebs, it's got to be aerobic respiration at this point. So, but glycolysis can happen both ways. We're going to break that glucose. We need a little help though. So we're about to break a bond. It takes some energy to break the bond, so we use ATP. So we do have to invest 2 ATP to break down that 6-carbon glucose molecule. And we see it, we steal those phosphates and now, or utilize the energy coming from those phosphates, and we lose the 2-ADP. Now we have two 3-carbon molecules called pyruvate. So we have the 2 pyruvate molecules. We're going to reduce, actually we're going to oxidize those. So we're going to oxidize those 3-carbon molecules a little further. Oxidation, remember, means that you're losing electrons. So where are we going to lose them? Well, we're going to actually gain some ATP in the process. So we generate 2 ATP from this 3-carbon pyruvate, and we reduce NAD plus to NADH. That means it's stealing an electron. It's being reduced, this is being oxidized, and so, hey, look, now we have an electron carrier. It looks very similar to the electron carrier we saw in photosynthesis, NADPH. The way that I remember is NADPH is the one in plants, or photosynthesis. And NADH is in cellular respiration. But same function, it is going to carry the electrons that it just stole from the pyruvate. And so now we have oxidized it to pyruvic acid. And it happens twice because we started with 6. Both of those 3-carbon molecules will go through the same process. So in the process, we generate NADH, which is our electron carrier. That's going to move forward and help us power the electron transport chain. And we generate a small amount of ATP. This is substrate level phosphorylation, where an enzyme directly is adding a phosphate to the ADP molecule. So it's not as energy efficient, but we can make some ATP, which is why anaerobic respiration will work. We do generate a little bit of ATP at this point. So before pyruvate will cruise on in and enter the Krebs cycle, we do a little modification of it. So it can enter the mitochondria, but before it enters the Krebs cycle, we oxidize it a little further and we break it down a little bit more. So we have pyruvic acid, that's the one that just came out of glycolysis, it's entered the mitochondria. We're going to oxidize it further, so we're going to steal an electron, we're going to reduce NAD plus again to NADH, so we've generated another NADH molecule, which is super for us because we need it to move forward in the electron transport chain and carry that electron forward for us. And we're going to release carbon dioxide. Also a good thing. So there goes another one of our carbons in the oxidation process. So we breathe out that carbon dioxide, and here it is. We're going to combine that molecule now. We lost a carbon, so we're down to 2 carbons. We're going to combine it with a molecule called coenzyme A to form acetyl-CoA. This is a 2-carbon molecule, and this is the one that will actually enter the Krebs cycle. This process happens twice because remember we had 2 pyruvic acids coming in from the single glucose molecule. So every round this will happen twice. So 2 acetyl-CoA, 2-carbon molecules are entering our Krebs cycle. And the Krebs cycle is where the magic starts to happen. So let's take a look. So in the Krebs cycle, we get a lot more CO2. So by the end of Krebs, we've gotten rid of all of our carbon molecules. We started with 6 carbons and glucose. We got rid of one of them with glycolysis. We lost a CO2, and now we're going to get rid of the rest of them. So CO2, and this is going to happen twice, remember. There are 2 of these coming in, so that's 4, and 2 in glycolysis. So we lost all 6 carbons by the end of the Krebs cycle. Where's all that energy going? We took this high energy glucose molecule, we're breaking it all apart. Where's all that energy going? It's going into making electron carriers, into reducing this NAD plus and FAD. We're going to reduce NAD plus and FAD to carry those electrons as NADH and FADH2. They're going to move forward in the electron transport chain, and that's the whole purpose of the Krebs cycle. The purpose of the Krebs cycle is to continue to oxidize, which means steal the electrons away. We're going to release the carbons as carbon dioxide. We're breaking down. In the meantime, we're stealing those electrons. Those electrons are going to power the proton gradient, the pumping of those protons out of the mitochondrial matrix so that they can come rushing back in oxidative phosphorylation. So it's all a big scheme, and it's working beautifully. So by the end, we have oxidized that, and we have lost our carbon molecules and gained a bunch of electron carriers. So let's kind of look at some of the summary here. So, like I said, these electrons are being extracted through this series of reactions. So we're building up NADH and FADH2 to power the electron transport chain. And you see that happening. We're losing carbon dioxide, gaining an electron carrier. Losing carbon dioxide, gaining an electron carrier. And that process continues. So, again, this is happening in our mitochondria. So in the mitochondrial matrix. It happens in the cytosol for prokaryotes. They don't have mitochondria. They don't have membrane-bound organelles. So it's happening in the cytosol, and they're actually going to pump protons outside their own plasma membrane, which is pretty cool. It is a repeated cycle. So it happens 2 times for every glucose molecule. And glucose is broken into 2 before entering, remember. So even before it entered, we had broken it down into pyruvate and then pyruvic acid before it can move into, and then ultimately into acetyl-CoA before it enters the Krebs cycle. 2 ATP are produced for each glucose per turn. So we've broken all of that down and we got 2 ATP out of it. Doesn't seem very efficient. Well, the efficiency is going to come in just 1 second. This was substrate level phosphorylation for those ATPs. So it is not very energy efficient. We only made 2. It seemed like a lot of work, but we were building up those carriers, those energy carriers as NADH and FADH2. So we're about to see how that is going to be also impressive as we make a lot of ATP. Okay, so, and as I said, we started with 6 carbons. We just lost 4 more during the Krebs cycle. We had lost 2 previously, and now during Krebs we're losing 2 more as CO2. In the process, we gained those wonderful electron carriers, 6 NADH, 2 FADH2, and we're going to use those in our electron transport chain. So let's take a look at that. All right, so the goal of the electron transport chain is to capture the free energy from those electrons that we just stole out of that carbon-based molecule through all those oxidations. We've reduced NADH and FADH2 to carry them in, and where else did we see this happen? It happened in chloroplast. Remember, we had photosystem 2 and then photosystem 1. In order to link the 2, we linked them through an electron transport chain. Photosystem 2 excited that energy in the electron, and then we had an electron transport chain, and that electrons got passed from molecule to molecule. Every time they did, that energy that was released helped power a proton gradient, which came rushing back in to form ATP, which helped power the Calvin cycle in photosynthesis. Now we're doing the very same thing. So if you understood that, you're in golden shape for right now. Now we're in the mitochondria, or the prokaryotic plasma membrane, if you're a prokaryote. And as we start to steal those or donate those protons back out, so the electrons from NADH are taken through the electron transport chain, we're building up, it's that energy that gets released helps power those hydrogens to come through. So we're powering the hydrogen, the protons to come back out of the mitochondrial membrane, and we're building them up, building them up, building them up. And every time that the electron carriers donate their electrons and pass them along through the electron transport chain, we release the positive proton, and that energy that was released from that electron is used to push those into a higher concentration gradient. And remember, that does take energy, and we're using that energy in the form of those electrons, which are moving from lower to lower energy states. They're all driven by this. These electrons are driven this way because they are very drawn to our friend oxygen, which, remember, we have to breathe in order for cell respiration to work. The oxygen is the final electron acceptor, and I think I'll get to that in the next slide. But all those electrons are drawn to this highly electronegative oxygen sitting out here. So electrons, electrons. And meanwhile, we're building up all that energy to form these protons. They're going to come rushing back in through ATP synthase. So let's take a look. So again, those electrons are being delivered by NADH and VDH2, ultimately moving to that oxygen molecule. They're very attracted to that. It is the final electron acceptor. If you don't breathe, what happens? It's going to shut down this whole process. There's no driving force for the electron transport chain. There's nothing pulling those electrons forward in such a way. So without oxygen, we can't make ATP, at least not sustainably. So big trouble. What was the final electron acceptor in photosynthesis? I don't know if you remember, but at the end of photosystem 1, after the electron transport chain finalized, it was NADP+. Okay, so a little bit different for plants, or for photosynthesis. In plants, their oxygen will also be their final electron acceptor during cellular respiration, but for photosynthesis, it's NADP+. All right, so as the electrons are passed, the protons are forming that gradient across the intermitochondrial membrane. It was the thylakoid membrane in photosynthesis, the plasma membrane in prokaryotes, all the same idea. We're building up a gradient of the protons on the outside of a permeable or a membrane, and they're going to want to move from high to low. They're going to come flowing back through ATP synthase. As they flow back through ATP synthase, again, they want to move from high to low. This is energetically very favorable. So they're moving from high to low, they're rushing back in. In the process, we make about 32 ATP per glucose molecule. So remember, Krebs, we barely made anything. Glycolysis, barely anything. But now we get 32 ATP, which is why we rely on that oxygen, because of this electron transport chain building up this proton gradient and the energetically favorable movement of those protons back into the mitochondria, and the beauty of the ATP synthase molecule. It's an enzyme. It has like a rotor on it. It very quickly attaches phosphate, so it's not substrate level. We call this oxidative phosphorylation. Very energy efficient, makes a ton of ATP. Yay! All right, this is an overview. I'm already at 22 minutes, so I'm not going to talk you through it, but I want you to pause it here and see if you can follow the movement. Start with the pyruvate from the cytoplasm, that's coming in from glycolysis, and follow point by point what is going on, okay? Take notes on this. This tells the whole story now that you understand, but I would draw this out. I totally would. This would help you out. The red lines are the electrons moving, right? So NADH is carrying those electrons. We have an electron transport chain. That's noted here. And ultimately, the electrons are being drawn toward that oxygen. The oxygens ultimately are going to combine with the hydrogens that are flowing back in, and we form water. That's one of our waste products, right? So those oxygens, once they've accepted that electron, will meet back up with the water, or with the hydrogens, because the hydrogens we know are attracted to that as well. We've got positive and negative, and we form a water molecule, and that's our waste product. So super cool. All right. I'm really trying to talk fast. Oh, this is the whole thing. Okay. So that's everything. Also, if you wanted all of those steps concisely written down, here you go. That's for you. I'll post these slides after you've taken your key up. Okay? So, but you'll have them for your notes, I promise. Anaerobic respiration. The big key there is eukaryotes form lactic acid. So we start with that pyruvic acid. We're out in the cytoplasm. Why do we form lactic acid? It actually helps us regenerate NAD+, which we need to initially break down the glucose. So it actually helps, and it makes it a bit of a cycle. That way we can cycle through this process. So we make lactic acid. It builds in our muscles. It doesn't feel very good. It's not sustainable long term. Yeast, bacteria, when they go through this process, they form alcohol. And one of the slightly humorous things to think of is, imagine if when we went through this process, we formed alcohol. That means when you're working out really hard, instead of building up lactic acid, suddenly you have alcohol now building up in your system, which would make for a completely different workout. So I suspect we might have more people exercising, but that's beside the point. Okay, so that's, I think, all I want to say. Oh, I'm sorry, I went 25 minutes. I really tried. Okay. Oh, here's some more notes. Just take them. CO2 is produced. Oh, CO2 is produced. I should say that. CO2 is produced during alcoholic fermentation. That gives you the fizziness in beer, helps bread rise. That's why bread rises. We have CO2 building up. It's a gas, and so it can rise. Wine, it's released, so you don't have that fizziness, but CO2 is a byproduct of alcoholic fermentation. In fact, we could, after the AP test, let's make homemade root beer. It's such a minimal alcohol content, it's less than like drinking some NyQuil. So you'd be fine. But it's really fun. I actually did it with summer school kids, and it was fun. So we can make homemade root beer. It's really fun. And you'll get to see that alcoholic fermentation and taste the fizziness of the CO2 that's produced. All right. There's these, if you want to take a look at them. They'll be in your notes. Make myself go away. They'll be in your notes for you to review. All right.